Hello, I'm Dr. Jose Floresci. In my last class on biotechnology, I discussed the tools of genetic engineering. We need two tools. One is DNA modifying enzymes, which include restriction endonuclease that cuts DNA, DNA ligase that joins DNA, and DNA polymerase that replicates DNA. Second tool is vectors. In the last class, I told you in detail about restriction endonucleases. Now let us look at the other two enzymes. Second one is DNA ligase that joins DNA. Ligation means joining. So what this enzyme does is joins two pieces of DNA. This enzyme is actually found in all bacteria. But for commercial purposes, we isolate it from E. coli. So that's the second enzyme. The third enzyme is DNA polymerase. That replicates DNA. DNA polymerase is actually used to make multiple copies of DNA. DNA polymerase is used in polymerase chain reaction or PCR. In PCR, we are heating the DNA to 95 degrees centigrade. We cannot use the normal DNA polymerase because it will denature at this temperature. Therefore, we use a special DNA polymerase called Taq polymerase, obtained from the bacterium Thermus aquaticus. The name Taq came from TAQ, Thermus aquaticus. This is a bacterium found in hot springs. So the DNA polymerase of this bacterium can function at 95 degrees centigrade. So we need three enzymes in genetic engineering: restriction endonuclease that cuts DNA, DNA ligase that joins DNA, and DNA polymerase that replicates DNA. Now let us look at the second tool in genetic engineering, namely vectors or carriers. See, you can actually transfer genes into organisms directly or using vectors. The importance of using vectors is that if you don't use a vector, only 1% of the cells get genetically modified. If you use vectors, 10% of the cells will get genetically modified. That is why we use vectors in genetic engineering. There are two types of vectors. One is cloning vectors. Second is expression vectors. Now, cloning vectors are used to make multiple copies of DNA. In the second step of genetic engineering, which is called amplification of the gene, we are making multiple copies of DNA. So, we use cloning vectors to make multiple copies of DNA. An alternative is polymerase chain reaction. Expression vectors are used to transfer genes into various organisms. I am going to give you examples. Let us look at examples of cloning vectors. A, PBR322. The full form is plasmid Bolivar Rodriguez 322. This is an artificial vector created by two scientists, Bolivar and Rodriguez. Second, Puck 18 plasmid, University of California. This vector was developed at the University of California. Bacterial artificial chromosome. Yak yeast artificial chromosome. Now these two vectors were not created for genetic engineering, they were created for the Human Genome Project. So these two were created for the Human Genome Project. If you look at the examples, one thing is very evident. These are all artificial vectors. So cloning means making multiple copies. Cloning vectors are used to make multiple copies of DNA. Now let us look at the second type of vectors, expression vectors. Now these vectors are used to make, these expression vectors are used to transfer genes into various organisms. Examples: A, TI plasmid or tumor-inducing plasmid. This is a vector used in plant genetic engineering. It is obtained from the bacterium Agrobacterium tumefaciens. This vector, TI plasmid or tumor-inducing plasmid, is used to transfer genes into plants. That is one example. Another example is retrovirus. You must have learned before, retrovirus contains RNA as the genetic material. So from the RNA of this virus, we have to make complementary DNA using reverse transcriptase. That is used to transfer genes into animals and humans. I'll give you an example. In the second chapter of biotechnology, we are learning about gene therapy, which is genetic engineering in humans. There, the vector used is DNA of retrovirus. So, that is an expression vector. These expression vectors are natural vectors. I'll repeat once more. Cloning vectors are used to make multiple copies of DNA. The most important vector we have to study is PBR322. Expression vectors are used to transfer genes into various organisms. And cloning vectors are artificial vectors. Expression vectors are natural vectors.